Hi and welcome to the 7.1 Inheritance PowerPoint. Is the specification if you want to stop it and have a quick read. I'm only going to do to this point here. So sex linkage, autosomal linkage and chi-squared aren't in this video. They'll be in the next one. Okay, these are the key genetic definitions. I'm not going to go through them all. You can pause it and read through them just to save time. Okay, so to begin with, we need to think about genetic diagrams. Now, you should have an understanding of this from GCSE already, and this should all be recap of GCSE, apart from maybe the way that this diagram is drawn, but these Punnett squares certainly should be a recap for you. So in this genetic diagram, we've got the parents. We've got parent A and parent B. Now, parent A has two dominant D alleles, and parent B has two recessive D alleles. In these genetic diagrams, you then have to show the gametes of each parent. So basically, each gamete gets one of these alleles. So this little d goes to this gamete here. This little d goes to this gamete here. Here, you've got big D to this gamete, big D to this gamete. So if the parents have two alleles, the gametes need to have one each, because obviously the gametes only have one set of chromosomes, whereas the parents will have two. You can then see that we've got the offspring genotypes here. Now, the offspring genotypes are determined from this Punnett square here. So these offspring are just the offspring from this Punnett square. In an exam, it's fine for you to draw a quick Punnett square to work out the offspring, but you can see that this one here has got a big D from this parent and a little d from this parent. This one here, big D, little d. This one, big D, little d. And this one is big D. Oh, little d there. So you can see the lines are coloured and you can see where each of the alleles are coming from for these offspring. And you can see that it marries up with this Punnett square here for the F1 generation. Now, F1 generation just means the first cross that we do with these parents. I'm going to show you the F2 generation as well. So the F2 generation is when we cross these offspring. So we take these offspring and cross them, and that's the F2 generation. You can also see on here that these all will have the dominant trait because the recessive trait will be masked by the dominant trait. That should be recap from GCSE as well. So the F2 generation is when we cross the F1 generation offspring. That's these guys. In this Punnett square here, I've done that. So you can see that you've got big D, little d, big D, little d. And then we'll cross it with this one, big D, little d, big D, little d. So here are the potential offspring. Now, when we cross two heterozygotes in a monohybrid cross, so this is called this all is called a monohybrid cross. When we cross two heterozygotes, you will always get a phenotypic ratio of 3:1. So you've got 1, 2, 3 will have the dominant phenotype, and this 1 will have the recessive phenotype. So the ratio of 3 dominant phenotypes to 1 recessive phenotype. Okay, so recessive traits, this is just the same principle, but we're looking at recessive and dominant traits. So cystic fibrosis is an example of a recessive disease, and it's caused when an individual inherits two recessive alleles from their parents. And if they have two recessive alleles, we refer to them as homozygous recessive. When we cross the two heterozygotes, you can see here that we've got big F, little f, big F, little f. The reason I used F's here rather than C's is because the capital F and the lowercase f different. So it's always good in an exam to choose, if they haven't given you a letter, to choose a letter where the upper and lowercase look different to avoid any confusion, because big C and little c can look the same. So we've got two heterozygotes crossed. So we can refer to these two parents as carriers. So if they have a dominant and a recessive allele, they aren't affected by cystic fibrosis, but they can pass this allele onto their offspring and they can be affected. So they carry the faulty allele, but they're not affected themselves. So we can see here that big F is two normal alleles. There'll be no cystic fibrosis. Big F, small f, that would be a carrier, so they're unaffected by the disease, again, here. So we've got 3 unaffected offspring and 1 affected. So we've still got 3:1 phenotypic ratio of unaffected to affected. and 50% of them are carriers as well. Okay, dominant traits. So Huntington's disease is an example of a dominant disease. Now, in a dominant disease, you only need to inherit one faulty allele in order to be affected by it. So you can either inherit one or two. But if you only inherit one faulty allele, then you will be affected. That's different to the recessive trait because one faulty allele, you're a carrier, you're not affected. So if we cross two heterozygotes here, you can see that we've got these three are now affected by Huntington's disease, and this one here is unaffected. So there's a 75% chance that the offspring will have Huntington's and only a 25% chance that they won't if both of the parents are heterozygotes. Notice as well that you can't be a carrier for a dominant disease because these two here, this one and this one, they have the recessive allele, which is the normal allele. In this case, it's the capital H that is the Huntington's. This is then masked by the dominant allele, and therefore they are affected by Huntington's. They can't carry it. Well, I mean, they are carrying it, but they're affected as well. So a carrier is unaffected by the disease but can pass it on. So no carriers for dominant traits. Okay, so codominant alleles. This is slightly different, and this should be new from GCSE. So codominant alleles in sickle cell anemia. They actually should have changed this. This should be HbA is the allele for normal hemoglobin, whilst HbS is the allele for sickle cell. Sorry, I didn't change that. You can see them here. So because HbA is normal, if somebody inherits HbA, they have normal hemoglobin. If someone inherits HbA/HbS, they have sickle cell trait. And if they have HbS/HbS, they have sickle cell anemia. People with sickle cell anemia have crescent-shaped or sickle-shaped red blood cells, and they aren't as efficient at carrying oxygen. People with sickle cell trait will have a few of the sickled cells, but still enough normal red blood cells to carry on as normal and not be affected by the disease. Interestingly, people with sickle cell anemia and sickle cell trait can't be infected with malaria. We've done that bit there. And then, okay, so here you can see that we're crossing somebody who has sickle cell trait with somebody who doesn't have sickle cell anemia. Now we end up with two, these are different as well, sorry. This should be HbS and HbA. I've not done very well here, have I? So the husband is HbA, normal hemoglobin. The mother is a carrier. So these two offspring here would have sickle cell trait, and these ones would have no sickle cell disease. So therefore, if you only have one parent with sickle cell trait, then the offspring can't have sickle cell anemia. You have to have two parents with sickle cell trait in order for them to have sickle cell anemia. And when we talk about codominant alleles, what we mean is when they come together like this, they make a brand new phenotype. So codominant alleles, a new phenotype is made. So rather than this phenotype just being normal hemoglobin as well, it's actually a new one called sickle cell trait. The ABO blood groups are also an example of codominance, which we'll have a look at in just a second. Okay, so blood groups is an example of multiple alleles, but also codominance. These are the blood groups here. So we've got AB, AB, and O. You can see the possible genotypes. So before we had HB as the hemoglobin for sickle cell trait. Here we're using a capital I for the blood type. And big A is obviously the blood group A. And big B, blood group B. Here, this is codominant because A and B have been inherited together to produce a new blood type of AB. And also, you've got this small I, which has neither the A or the B on it. And if you inherit two of those, then you would have blood type O. Now, IA and IB are codominant, so they produce type AB when inherited together, but they're also dominant over this small I. So here, if you inherit IA and a small I, then you would be type A because this A is dominant. This B is also dominant over the small I, so this person would be type B. Here you would be AB. This is type O because there's neither A nor B. We've also got here an example of the cross. So if somebody is group A, IAIO, the gametes, they've used IO here instead of a small I, and that's fine. Gametes are IA and IO. Again, this should be B. Sorry guys, I've chosen some really crap diagrams. This should be IB or IO. And then here you can see that if a type A parent and a type B parent are crossed, you would get 25% chance of AB blood type, 25% of A blood type, 25% chance of B, and 25% chance of O type blood. The biggest thing to remember here is that you have to use the I's because you don't get all of the marks if you don't use the I's for blood groups. Oh, look, I wrote that there. I think that's it. Oh, it's not it. This is dihybrid crosses. So this is the inheritance of two different traits at the same time. So you can see here that we've got Y's and R's. So Y stands for yellow peas, small y is green peas, big R is round peas, and small r is wrinkled peas. So it shows how more than one characteristic is inherited. So what happens is we end up with gametes. Now, there should be 4 gametes here. I think it's easier to draw it out with 4 gametes. And they would all be big Y, big R. And these ones would all be big Y, little r. When you cross them, you find that all of the offspring would be big Y, big R, little r. So you can draw this Punnett square, this 16 offspring Punnett square, for this cross if you want to, just to prove that all of the first generation, the F1 would be big Y, big R, little r. These would be the gametes here for this individual. So you take big Y and big R here. Then you've got big Y and little r here. Little y, big R here. Little y, little r here. Now in the second generation, we're going to cross 2 of the first generation offspring again. So you can see here that we've taken those gametes here and we've taken them here, and then we do all of the possible offspring, and there's 16 possible offspring. Now in an exam, it's really important that you write out the genotype and you write out the phenotype within these boxes, because that gets you all of the marks. You can also see here that once you've done that, you've got 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled. That gives us a ratio of 9 to 3 to 3 to 1. Now that's our classic dihybrid Mendelian ratio for crossing 2 heterozygotes, in the same way that when you cross 2 heterozygotes in a monohybrid cross, the ratio would be 3 to 1. So the standard ratio always 9 to 3 to 3 to 1 for crossing 2 heterozygotes. And I've just said make sure that each gamete has 2 alleles. The gametes have 2 alleles, and the potential offspring all have 4 alleles, 2 of each letter for dihybrid crosses. And that's all for today.